Hi, I'm Elizabeth Redcay. I'm an associate professor of psychology at the University of Maryland. And I'm really excited to be part of this series of talks on naturalistic imaging. Many of the speakers in this workshop have really laid the foundation for some of the work I'm going to discuss here, where I'll be talking about the social development of the social brain and individual differences using naturalistic imaging approaches. So I'm going to start this talk with the most important slide, which is the acknowledgements. Because of course, none of this work would have happened if it hadn't been for the members of my lab, and I will highlight the key individuals throughout the talk. And of course, I want to say a big thank you as well to the children and families, as well as the students who participated in this work, and to NIH for funding these projects. So as we've all become acutely aware over the past year and a half, social interactions are important. From the first minutes of life, they shape who we are, what we attend to, and our likes and dislikes. The who, how, and why may shift over development, but they continue to play a central role in our lives. And this pandemic has brought into sharp focus that reduced or impoverished social interactions and social relationships can have devastating negative effects on mental health, physical health, and well-being. So the pandemic is temporary, but many individuals live with chronic difficulties navigating social interactions and forming close personal bonds. This includes neurodiverse individuals, such as those on the autism spectrum. So a central question of my research program is what cognitive and neural factors underlie the typical and atypical development of social interaction. Much of the work addressing this question has really pointed to the social brain. So this is a little bit of an ill-defined term, but generally involves regions associated with theory of mind or mentalizing, such as these regions in blue, as well as regions associated with reward and affect processing, including these in red, and regions in green here associated with action perception. And in particular, much theoretical and empirical work has suggested a key role of theory of mind. That is our ability to perceive and reason about another's mind. So here I'm showing a meta-analysis that's demonstrating key regions associated with a diverse array of theory of mind tasks. And it highlights this network of regions, often referred to as the mentalizing network, a network that's overlapping with the default mode network. But the majority of tasks and approaches used to study theory of mind have used relatively impoverished social stimuli that are divorced from their social context. So just to give you an example, a very common test of theory of mind is known as the reading the mind in the eyes task. So in this task, a participant comes into the lab and sits in front of a computer screen or goes into a scanner, and is presented with a slice of just the eyes and asked to pick which label matches how they are thinking or feeling. So I'm going to encourage you, the viewer, to just first take a moment and reflect on whether or not this person is feeling friendly, annoyed, sad, or disgusted. So when I've been able to give this particular slide to live audiences, typically people respond that they think that she is feeling sad. And in fact, this kind of highlights that there are two limitations with this typical traditional approach for assessing theory of mind. One is that real-world social processing is complex and dynamic. So you are rarely presented with just eyes when you have to make an inference about someone's mental state. But instead, you have the rest of their face, their body, and that the rest of the social context that helps you make that kind of inference. Also importantly, you have the temporal context. So we don't encounter social stimuli as static slices in time throughout our lives, but instead, you know what happened just prior.
I guess a cup of coffee would be great.
Wait a second. Oh, you're spotted. Another addict. Oh, gotta love the box.
What?
It's like a slang for Starbucks. They're all over the place. Oh, man. That place is like the promised land to me. What a business model, too. Oh, too bad we don't have the...
So with this context, it then becomes readily clear that she's not feeling sad. She's instead feeling annoyed after this somewhat awkward interaction with Michael Scott from The Office. A second limitation, though, is that real-world social processing involves social interaction. So we typically aren't presented with social stimuli just via video screen, but instead we are often presented with a social stimulus in the context of a social interaction. And so we don't have the luxury of just reflecting on what that person's thinking. We need to make that inference more rapidly and in real time. And the extent to which we are accurate in making that inference can have real world consequences on how the conversation is going, as well as in the development of our relationship. So in this talk, I'm going to focus on naturalistic approaches that address these limitations. So that is approaches that either engage the viewer in a social interactive context, or they present the viewer with dynamic, complex, and contextually embedded social stimuli, or in some cases, address both of these limitations together. So a growing body of work has used these naturalistic social interactive approaches and is demonstrating that there are differences in brain function between interactive and observer approaches. So the observer approach, also known as a third person approach, is where you present a person in the scanner with a image or video of a non-interactive social stimulus. An interactive approach instead engages that participant in a real-time social interaction. And this work is showing that there's evidence for greater functional connectivity within and between regions of the social brain when engaged in that interactive context compared to the observer approach when you're just viewing that social stimulus. In another example, work from my lab has demonstrated that the mentalizing has demonstrated another contrast. So specifically, the mentalizing network is engaged when reasoning about a character's mental state. So here you see in these non-interactive contexts, when reasoning about mental states, you see engagement of this mentalizing network. But when you're doing similar kinds of reasoning about a person, but it doesn't involve mentalistic information, then you see significantly less engagement of this mentalizing network. However, when in an interactive context, that is when you're directly interacting with a social partner, you see that the mentalizing network is engaged similarly, either when you're making judgments about that social partner's mental states or when you're making judgments that don't involve or require reasoning about that person's mental state. So these findings highlight that appeals to ecological validity are theoretically important. So that is what we learn about brain function differs depending on that context. So these examples and much of my prior work have really focused on that interactive context, but examining social stimuli embedded within a complex dynamic context encountered in the real world can also reveal novel insights about brain function. So just to get back to that first question I posed that really drives much of my lab's research. What are the cognitive and neural factors that affect the typical and atypical development of social interaction? So in this talk, I'll discuss how we've used naturalistic approaches to address this question. First, I'll discuss insights that we've gained about the development of the social brain, focusing on how the brain processes social stimuli embedded within this rich social context, and ask how does this change with age and how is it related to individual differences in social abilities. And then the second part, I will discuss work using naturalistic imaging approaches to examine how social interactive context modulates brain function and predicts individual variability in social behavior. And I'm going to focus on two key aspects of social interaction. So these are social sharing or sharing attention or joint attention with another individual, and social reward, specifically here, receiving a positive response from a peer. So the majority of studies on the development of the social brain have used constrained task-based paradigms or unconstrained resting state paradigms. And a general pattern across these is that the brain undergoes progressive functional specialization. So for example, in a task paradigm, you might present an individual with social versus non-social stimulus, like face versus house. And what you would see is that in younger children, there's a more diffuse response. So less selectivity in voxels and a lower amplitude response across multiple voxels. But with age, that becomes more focal. So there's a greater intensity within a more focal set of voxels. And while this work is important for precise stimulus control, and I use these approaches in my own work as well, there are limitations in our understanding of the development of the social brain. So primarily, as mentioned previously, it's unclear the extent to which these findings generalize to the complexity of social processing that we might encounter in everyday life. There's also limitations on the age at which children can be scanned when they have to engage in a task and remain still, or unconstrained rest, which also presents a challenge to remaining still. So we wanted to know how, and in particular, the early childhood, when children are around 4 to 6 years of age, is a really interesting time in the development of social abilities. So this is a time when they, in particular, pass the canonical false belief test, sort of a hallmark of theory of mind development. So we wanted to ask the question of how is the brain specializing to complex social stimuli during this exciting time? So to address this question, we leveraged prior work showing that movies allow for greater high quality data from young children. So that's Tammy Vanderwall and colleagues have shown that, as well as that movies maintain their ability to examine functional specialization to the social stimuli, even when embedded within a complex video. And specifically, when watching a video, you can use inter-subject correlation approaches to look at the reliability in how the reliability in the response to a stimulus changes with age. So with inter-subject correlation, we look at the time series of a given voxel and see how correlated that time series is to the average of the rest of the participants within the group. And what we would expect is that younger ages may show less consistent responses to stimuli, but it becomes more focal and selective with age. As the regional bold response becomes itself more focal and selective, you would see that more consistency or reliability across participants. So this inter-subject correlation method during movie viewing will allow for the examination of functional specialization to social stimuli that are embedded within this rich and dynamic social context. And to do this, we presented participants aged 4 and 6 years, as well as adults, with a clip from the movie Toy Story. And we identified a clip that really elicited a lot of inferences about characters' mental states. And here I'm presenting just the participants who provided usable, that is low motion artifact data. So it's 23 4-year-olds, 23 6-year-olds, and 21 adults. And this work was really led by my former graduate student, Dustin Marcheski. So when doing a direct comparison of inter-subject correlation between the adult and the child, we see many regions that show higher reliability, higher ISC in the adult as compared to the child. However, because viewing is unconstrained, we also did an analysis where we used a regressor of the bold time course from the frontal eye fields as a way to account for some of the differences that may be due to saccades or other attentional differences that we might expect with age. And so this is this controlling for attention analysis. And here we still see that there's greater inter-subject correlation, largely within some of these social brain regions like the temporal parietal junction and superior temporal sulcus. So this is evidence for support for our hypothesis that functional specialization increases with age, but we show this using this novel approach using movie stimuli, showing more consistent patterns elicited in adults compared to children. And in order to look at age-related effects between 4 to 6 years of age, we used a measure called neuromaturity. That is each child's time series correlated with the adults. And here we see a significant increase in social brain regions, including bilateral TPJ, though only the left TPJ remained when we controlled for attention. So to display this change more clearly, what I'm showing here is all pairwise correlations within that left TPJ region. And what you can see is that the correlations to the adult increase from 4 years to 6 years into adult. So the TPJ is a key node within the social brain, particularly in reasoning and about beliefs. And this is an ability that does show significant change within this early childhood age range. However, the TPJ is a multifunctional region, and it's also involved in attention, memory, and language processes. And so these data can't speak to why we see this increased reliability between 4 and 6 years of age and whether or not it's associated with social advances. So for our next study, we aim to examine functional specialization and relate it to behavioral performance on social cognitive tasks. So also around this time that we're thinking about this study, my graduate student, former graduate student, Dustin, was reading and excited about work from Nuri Hassan's lab and colleagues demonstrating that naturalistic FMRI approaches could be used to map out a cortical temporal hierarchy. And regions overlapping with this default mode network were more sensitive to information presented over long time scales, like a narrative or a movie. So in other words, the way that a particular stimulus is processed will be affected by prior context. And the window of time that can influence that process is that region's timescale. So these regions within the default mode network, which I mentioned, some of which are overlapping with the mentalizing network, have long temporal receptive windows. And we thought about these long time windows that they might be especially important in aspects of social cognitive processing. So because it's important to develop a model of one's social partner's mind or of a character's knowledge in a story in order to accurately and efficiently make predictions about their behavior based on their mental states. And most studies of temporal receptive windows have used social stimuli, but they hadn't really explicitly linked this long timescale processing to social processing. And also there was not information about how these temporal receptive windows develop. So in this study, we had two primary questions. How does long timescale processing change with age? And are these differences related to social cognitive development? So in this study, we used a slightly older age range, looking at children within middle childhood, 6 to 13 years of age. This is still a time of interesting social cognitive developments, particularly in more advanced aspects of theory of mind development. And it allowed us to also scan for a longer period of time that we needed to be able to examine these questions of the development of timescale processing. So again, usable data from 31 children aged 6 to 13 years of age and 24 adults. So we presented children with a 6-minute clip of a children's TV show known as called Little Lunch. So I'll just show you a clip.
But you never let anyone else have a turn. You think you own the monkey bars.
Mara definitely thinks she owns the monkey bars.
I thought she owned the monkey bars.
I don't think I own the monkey bars. I don't know why anybody would say that. Get off my monkey bars.
Tomorrow's dad buy the monkey bars at a shop and bring them to the school playground just for tomorrow to play with?
Okay, so we chose this clip both because it had these short scene cuts and it also required thinking about characters' mental states in order to follow the plot. So that is the intact video. And in order to isolate long timescales, we compared it to a scrambled clip in which the order of the scenes were scrambled, but it maintained those the scene cuts. So I'll give you an example of what that was like with the scrambled video.
You are really, really annoying. But you never let anyone else have a turn. You think you own the monkey bars. I don't really care about cricket nets, but I do care about a match learning opportunity. I was hurting all over, but I wasn't going to let go. Nothing was going to make me let go. Almost nothing.
Get off, Melanie. It's my turn on the monkey bars.
Okay, so here, when you're viewing that, you each scene makes sense, but you don't have the prior context to really interpret the scene and the character's intentions. And here I'm showing the same video just for the talk, but in the actual scan, the intact and the scrambled episodes were different, and that was counterbalanced across participants. So after they watched those videos in the scanner, they answered some comprehension questions following the scan. So these comprise mental and non-mental comprehension questions and were done for both intact and scrambled videos. And to look at changes in long timescale processing associated with social cognition, we focused on regions of interest that are identified in that meta-analysis that I showed you previously of theory of mind processing. So these included regions of the precuneus, DMPFC, and TPJ. And then we had an auditory region, which was really a control region that we didn't expect to be associated with long timescale processing. First, I'll show data from an approach to examine long timescale processing by looking at the power spectra of the bold signal while viewing the intact episode. So we were here interested in the proportion of the spectral window that's comprised of low frequency power, as that would be indicative or suggestive of long timescale processing. Here, the adults are in the darker color and the children are in the lighter colors. And in the TPJ, we see that there's significantly greater low frequency power in the adults compared to the children. So again, that's indicative of age-related changes in long timescale processing within this TPJ region. And a second method to examine long timescale processing is by comparing ISE in the intact compared to the scrambled episode and looking at age-related differences, so comparing adults to children in this contrast of intact compared to scrambled episodes. So in the middle is the whole brain analysis, and the graphs are depicting ISE extracted from the regions of interest from those theory of mind ROIs. And in the orange is the ISE from the intact episode. In yellow is the ISE from the scrambled episode. Adults are on the left side of the graph. Children are on the right side. And what we see here in two of these theory of mind regions, the DMPFC and precuneus, there's significantly greater ISE in the intact compared to the scrambled episode in the adults, but you don't see that effect in the children. So these data, again, are suggestive of age-related differences in long timescale processing within these regions associated with social processing. And behaviorally, we see that these timescales also affect social cognitive comprehension. So accuracy was worse for mental questions when presented in the scrambled condition, but this did not affect questions that were not about mental states. So finally, to examine whether neural processing related to these behavioral effects, we looked at whether neural maturity in the intact versus scrambled episodes was associated with accuracy for mental compared to non-mental questions. And we see here that neural maturity of the DMPFC was associated with greater comprehension, accuracy for mental compared to non-mental questions. So this might have to do with the DMPFC's role in tracking intentions between characters or building up models of a character's mental state. But importantly, we didn't see any of these significant effects within our a priori theory of mind ROIs, not even the DMPFC ROI. So we're still considering this finding preliminary. Okay, so in sum, we showed that long timescale processing shows age-related differences. You see increased low frequency power in the adults compared to the children, as well as greater response reliability in the intact compared to the scrambled episode. Behaviorally, timescales affect social cognitive comprehension in children. And the DMPFC maturity during long timescale processing may relate to social cognitive comprehension. So overall, we've demonstrated that movie stimuli can provide new insights into the development of the social brain. And we've begun investigating how these changes may be related to individual differences in social abilities. But recent work has demonstrated that movie stimuli really offer an excellent opportunity to look at individual differences, given that they may provide more reliable markers and are easier to collect from large samples, particularly easier for children. And conveniently, there are now several publicly available developmental data sets of movie viewing with much larger samples in which examination of individual differences can be leveraged for a more robust analysis. In particular, here I'm just highlighting data from the Healthy Child Mind Institute, the Healthy Brain Network, as well as Partly Cloudy, which is work done by Rebecca Sachs and Hillary Richardson and colleagues. And this Partly Cloudy data set is it's a 6-minute short animated video from Pixar that is a really nice way to look at the development of the social brain, because it has a lot of scenes that really elicit theory of mind reasoning. And in addition to having this short, engaging clip, that data collected from these children, they also collected data on behaviorally looking at an explicit theory of mind battery. And there's over 150 children ages 3 to 12 who participated in this movie scan, contributed data from movie viewing, as well as data from this behavioral battery. And the behavioral battery, I'm showing an example of the false belief task, but it was in fact a battery that really was designed to probe individual differences in the types of theory of mind abilities that are developing over that 3 to 12 age range. So it had more sophisticated and complex aspects of theory of mind reasoning. So a current postdoc in my lab, Oliver CA, has leveraged this data set to examine individual differences in social brain development. And in particular, he took advantage of the fact that movie stimuli allow one to examine different aspects of the bold signal. So during movie viewing, the bold time course is comprised of stimulus-related signal, intrinsic signal, and non-neuronal noise. Within subject, functional connectivity can be driven by any of these components. But intersubject functional correlation can capture what's, because it captures what's shared between individuals, can really isolate this stimulus-related signal. And so ISFC, or intersubject functional correlation, allows for a network approach like functional connectivity by correlating each node of the child to all of the other nodes from the adult. So using these approaches, Oliver asked which provides better information to predict social abilities. And he used the Social Brain Atlas for regions of interest, and he extracted time courses for those and calculated the intra-subject functional connectivity, as well as the intra-subject functional correlations. And within these data, so we had a measure of FC and ISFC for each individual, and he looked at how those measures could predict their behavioral abilities, or the theory of mind score. To do this, he built a partial least squares regression to identify this brain behavior relationship. So briefly, PLSR is a multivariate regression technique that finds the principal components that maximize the covariance between the brain signal, that's the FC or ISFC, and the output behavioral data, that is the theory of mind score. Here's a toy example of the first PLSR component extracted, and note that the first component mostly points to the X1 and X2 direction. Then that would be capturing fixed features that are most predictive to Y. A leave one out cross-validation was then used to find the optimal dimensionality of latent variables, or the number of components, by minimizing the predictive mean absolute error. And then this MAE was used to compare model performance between a model with just functional connectivity, with just intersubject functional correlation, or with the combination of functional connectivity and intersubject functional correlation. And what he found is that even across a number of different components, the joint combination of ISFC and FC was best at predicting social cognitive abilities. And importantly, this effect held even when having the equivalent amount of data across all three models. So when randomly sampling to match the dimensionality across all models, the model performance with this joint connectome was still superior to either of these alone. And just shown another way, you can see that the correlation between the predicted theory of mind and the true theory of mind is really highest for this combination of functional connectivity and intersubject functional correlation. So in ongoing work, Oliver is examining which features or nodes best predict social abilities that can inform our understanding of how changes in the brain networks and naturalistic settings underlie developing social competence. So just to summarize, we've used naturalistic stimuli, particularly movie data, to provide novel and complementary insights into the development of the social brain. So we show that regions of the default mode network, especially the left temporal parietal junction, show increasing functional specialization in early childhood. In middle childhood, DMN regions become increasingly sensitive to information presented at long temporal scales. And this development may be associated with social cognitive developments. But finally, movie data can allow for the examination of how social brain networks develop using complementary information representing signal and intrinsic network responses. And this combined information is a more robust predictor of individual differences in social abilities. So in the second half of this talk, I will discuss work using naturalistic approaches, including social interactive methods, to examine how key social interactive behaviors are supported by the brain and relate to individual differences in social abilities. Okay, so in part one, I discussed how the brain processes social stimuli when it's embedded within a complex naturalistic social context by using movie stimuli. But you, as the viewer, are also typically embedded within a social context, and that context, that is others in your environment, can have significant impacts on how you process those complex social stimuli. So this phenomenon, known as shared attention or joint attention, has typically been examined or studied using very simplified stimuli and paradigms that really don't reflect the real world experiences of shared attention, which are dynamic, complex, and emotional. So in the current study, we aim to examine the neural correlates of sharing attention when attention is directed to dynamic, complex stimuli that vary in their emotional content. And this work was led by a postdoc in my lab, Sarah DeZura, and a graduate student, Junaid Merchant. So in this task, we again manipulated the social context. So they were either viewing a movie with a social partner who was actually a Confederate, but they believed was a social partner, or they were viewing it on their own, the solo condition. And the videos either were positive, neutral, or negative, as rated by a separate study of raters who assessed these videos. So they would first have a behavioral session with their Confederate where they just did some interactions and got to know each other. And then later would go to the MRI scan and the participant would participate in the MRI study while the Confederate was outside in the control room. And the participant believed the Confederate was watching the videos at the same time as they were while in the scanner. Specifically in the joint condition, they got a cue that they'd be watching videos together. And they just had this cue on the screen to reinforce the idea that when the Confederate was looking at the video, then they were watching the video together in real time. However, when they were in their alone condition, the Confederate would be facing away from the video to remind them that the Confederate's watching a different video, but that than they are watching. And they believe that in fact, these videos were videos taken in real time from the control room. So after each video, which lasted about 20 to 30 seconds, they responded to the question, how alone or together do you feel from a scale of 1 to 9? And how positive or negative do you feel, again, from a scale of 1 to 9? They then received the response from their partner of what they picked, as well as in the alone condition, they just received the kind of average response of what participants tended to pick. And they also had an opportunity between runs to talk with each other. So this was really meant to kind of reinforce this idea that you're watching this video with this person and you're sharing that experience together. So our ratings from within the scanner demonstrate that as we expected, that negative videos were rated as more negative, positive videos were rated as more positive. But there were no differences between the joint and solo conditions. So this went against our hypothesis that sharing with another person might influence your judgment of that video. However, what we did see was that subjects did report feeling significantly more together with their partner in all joint conditions compared to the solo conditions. They also reported feeling more together during the positive condition compared to other emotion categories. And this effect was driven specifically by the positive joint condition. So participants feel more positive when watching with a person, and this is greatest when they're sharing attention together on a video that elicits positive affect. So with these data, we can take multiple different fMRI analyses approaches given the design. And this is work led by Sarah. So first we can use a more kind of traditional approach of the general linear model, where we can look at the average responses across different classes of stimuli. It's really best for looking at questions where the stimuli are exhibiting a sustained response and are really kind of consistent across the different, across the trials. But ISC can identify dynamic stimulus-related responses that are reliable across individuals, even if they differ between trials. And this is ability to detect that idiosyncratic temporal structure of the more naturalistic videos is especially important in understanding real-world emotion processing as emotions unfold and fluctuate dynamically over time. So I want to just highlight an example of that by showing some videos that we presented to our participants in the scanner. So these are both examples of positive videos. And here's another positive video. And so importantly, while these were all rated separately as consistently eliciting positive feelings among viewers, they have very different temporal structures, even within emotion relevant regions based on kind of when and how those emotions would be perceived. So first to look at the effects of social sharing, we found that in both the GLM and ISC approaches, the DMPFC was engaged more for shared compared to solo videos. and that's independent of the emotional condition. So this is consistent with a large body of literature showing the importance of this region to joint attention. And these data, though, isolate that phenomenon, the phenomenon of that experience of sharing with another person without conflating the coordinating attention or responding to gaze cues. We also found interactions between social and emotional stimuli. So with the GLM approach, there was an interaction in the ventral medial prefrontal cortex. And this is an area known to be involved in both social and emotional stimuli. And we see that this region is engaged more in the joint conditions for the emotional stimuli specifically. And this region was not seen in the ISC analysis, and that's consistent with prior work showing that this region reflects more endogenously driven activity that may have a more idiosyncratic profile across participants. In the ISC data, we saw an interaction effect within the amygdala and the STS. So specifically when viewing movies alone, the amygdala showed more similarity in the negative movies compared to the neutral or positive. And this was within the amygdala, a region that's involved in emotion processing, but in particular in processing highly salient stimuli. So it may be that these negative stimuli led to kind of a more similar response profile across participants. However, when in a joint context, we actually see the opposite. So we see greater similarity in the amygdala when individuals are viewing positive videos compared to the negative or neutral. So together, these data are consistent with work showing that affective stimuli may lead to greater neural alignment between individuals, but they suggest that positive events may be more salient when together and negative events may be more salient when alone. So those findings that I was showing were all at the group level. And as a group, people engaged the MPFC more for shared events and show differential effects of social context when viewing emotions within regions associated with salience processing like the amygdala. However, the extent to which they are influenced by this social context may vary based on their social abilities. For example, based on their motivation to engage with others or social reward, their propensity to think about others' mental states, and their social behaviors. And each of these measures has different subscales. And so we can look at an individual's social profile by looking at their responses to each of these subscales. So for example, this purple person might have a particular profile based on how they respond to these different measures. Whereas the orange person might respond a little bit different on the measures, but overall, if you correlated these two profiles, you'd see that the purple and the orange individuals are quite similar. The green individual, however, might have a very different response to each of these questionnaires, thus having them be more dissimilar in their social profile. And so this is work led by Junaid, and he showed, and what he did was used intersubject representational similarity analyses to look at how social similarity is related to neural similarity. So in particular, this subject by subject matrix is created for the intersubject social similarity by correlating these behavioral profiles for each pair of participants. And then the intersubject correlation matrix is created for each ROI by extracting the time course. Here we used the Shen parcellation with 268 parcels, and show that, and then could get a matrix of each individual's pairwise correlations across each of those parcels. So then you can calculate the correlation between the social similarity and the ISC. And that comparison can help us to understand the factors that might be associated with variability in ISC in these higher order social brain regions. And so what we find is that when you look at how similarity of social profile is related to similarity in neural response or the intersubject correlation, there is an effect in these some of these social brain regions, that's midline regions, DMPFC and precuneus, as well as amygdala, within the joint condition, but we don't see these effects in the solo condition. However, an important caveat is that these regions do not survive correction for multiple comparisons using FDR when we're correcting for the 268 parcels used from the atlas. So we wanted to drill down more within the emotion conditions because we saw in the group analysis that there were these differential effects depending on if you were sharing attention on a positive video or a negative video. And indeed, what we see is that when you look at the relation between similarity in the social profile compared to the ISC, we do see an effect within these regions associated with salience. And in fact, the purple outline of the region indicates that that region did survive multiple comparisons correction using FDR. So this anterior cingulate and insula regions are associated with salience processing. And we see that the extent to which someone is similar in their social profile means that they are more similar in their neural response within these regions. And we did not see that effect within the negative videos. So there was no effect either for either joint or solo conditions within the negative videos. And we want to unpack these effects in that joint positive condition by looking at profiles within the behavioral measures. So here, just showing back to the all emotional processing, we see that the effects of social context are present within the amygdala. So that is the similarity in the social reward profile is related to inter-subject correlation within the amygdala. And we don't see this effect within, and we do see this effect within the positive conditions. So there's a greater effect within the amygdala and MTL regions when in the joint positive conditions, but not in the solo positive condition. So these data suggest that the effects of social context might vary based on similarity in sociability, particularly in individual differences in the social reward profile, affect how similar processing is within the amygdala. And taken together across group and individual difference results, we see that positive emotions might be more salient in joint contexts, and that the salience of these may vary based on individual differences in social abilities. So I showed that sharing with others can affect how emotional stimuli are processed, and individual differences in reward may contribute to that. So I want to close on a study that's investigating whether individual differences in neural sensitivity to social reward can predict social behavior in a real-world peer interaction. And this is work led by my graduate student, Catherine McNaughton. And she asked whether or not reward network activation can predict interaction enjoyment. And so that's interaction enjoyment between two peers in a real-world social interaction. So to examine neural sensitivity to social reward, we used a social interactive FMRI task that was designed to probe the neural bases of social reward. And we collected data from 71 children. So that's, again, usable scan data, so at low motion artifact. And these children were 8 to 14 years of age. So just to briefly orient you to the task, we had participants believe that they were engaging with a peer who was in another lab, also participating in a similar experiment. And they would share information about themselves, like, I like cookies. So they would respond yes or no to that. And in the peer conditions, they would either get a response back from the peer, and the majority of these trials were the positive response back, me too, and those are the ones that we analyzed. Whereas if the peer was away, they got a response that the peer read their message, but they weren't available to respond. In the computer conditions, so this is our non-social control, they shared information about themselves, but this time it was just to a computer, and they were told that the computer was just going to spin a wheel at random that either matched or did not match their response. And then in the control condition for the away, they were told the computer sometimes is disconnected. And for more detail on this, you can take a look at this Warnell et al. 2018 paper in Developmental Science. So the analyses that I'm going to talk about will just focus on this reply period as the event of interest. And the reason for this is that we see in previous work in our lab that the ventral striatum shows is robustly engaged to receiving these peer engaged message. So this me too from the peer compared to these control conditions. And so we wanted to know, is this effect within ventral striatum for the specific contrast of social reward? Can that predict how much an individual will enjoy an interaction with a real peer, a real participant in the lab in a naturalistic social interaction? even years later. And so we brought these participants back with an average of 2 years later, and we had them pair up and engage in a social interaction that was somewhat ranged from structured to unstructured. So there's a getting to know you task, as well as some more semi-structured tasks. And then we asked them afterwards, on a slider scale, if we bring you back for other studies, how much do you want to interact with this same participant again, or get matched up with a different participant? And we also had them fill out an interaction quality questionnaire. It's just a 6-item questionnaire about their interaction. And what we found is that that sensitivity to social reward within the ventral striatum is related to their response to how much they want to interact with that peer, again, years later. So this is ventral striatum activation to social reward with a peer within an FMRI context is related to how much they want to interact with this novel peer in a totally different setting. And we found a similar effect within a marginal effect in this relation between ventral striatum activation and interaction quality of that interaction. So together, these findings show that the ventral striatum sensitivity to peer interaction significantly predicts self-reported desire to interact with a novel peer, and it marginally predicts self-reported interaction quality. And in a larger sample where we're currently writing these data up that includes children with autism, we see also that the ventral striatal response to the social reward is predictive of both interaction quality and desire to interact with that peer again. And that suggests that similar mechanisms might underlie this social enjoyment in both children with and without autism. To conclude, we use naturalistic imaging approaches to examine the development of the social brain, the effects of social interactive context, and how individual differences modulate these effects. Using movies, I showed that there's increasing functional specialization to complex social stimuli with age in regions associated with social processing, like the TPJ. And we also showed that some of these social brain regions show increasing selectivity for processing requiring long temporal receptive windows. And further, individual differences in social cognitive abilities in children are best predicted by the combination of inter-subject functional connectivity and inter-subject functional correlations during movie viewing. These naturalistic approaches also provide insight into how the brain responds during social interaction. So I demonstrated that positive emotional context influences feelings of togetherness and leads to greater neural similarity during movie viewing with a social partner within the amygdala. And individual differences within one's behavioral social profile, particularly their social reward profile, are related to greater neural similarity during movie viewing with a social partner, again, within the amygdala. And finally, individual differences in the neural sensitivity to social interactive reward within the ventral striatum, and that social reward is receiving a positive response or reply from a social partner, predicts how much a child will enjoy a peer interaction, even measured years later. So together, these findings demonstrate that naturalistic stimuli provide novel insights into our understanding of social development and social interaction. Traditional approaches with precise experimental control should also be used in combination with naturalistic imaging approaches. And together, with these approaches, we can gain a full understanding of how the brain supports the typical and atypical development of social interaction. Thank you for listening.